Hi again. Today we're going to look at replication, and we're also going to look at the hierarchy of genetics. So replication is defined as copying DNA. Okay, so replication, copying DNA. If I ask you to replicate something, that kind of makes sense, right? If I ask you to replicate, I'm asking you to copy it, right? If I ask you to replicate what I'm doing on the board, you're copying what I'm doing on the board. Now, it's done in preparation for mitosis, right? And we learned in the last unit that mitosis during the S phase of interphase actually is when you do replication. That's where you copy the DNA, and that's to get ready to do mitosis. Okay. All right, so we're going to use a particular style of drawing for this next bit, where you actually draw out how replication works. Replication is really a very simple process, and it's easy to kind of draw it here and model it. The first thing you have to understand, though, I've drawn kind of a little DNA model here, and I want you to understand that instead of putting all the phosphates and sugars into the backbone, just to make this easier, since from here on out what we're really concerned with is the code that's on the molecule, not so much the backbone, because the backbone is just going to repeat. I've drawn the backbone as these lines here. Okay, so these are the backbones. Okay, so those two lines are the backbone. And now we're going to focus on the individual letters of the DNA code that we see in the middle of the molecule. All right, so let's look at how replication happens. Now that we've looked at how we've drawn it, what I've got is step one. Here's the DNA that I drew just on the previous slide. Okay, so there's step one. And what happens in replication is, because remember right here, the nucleotides, the bases of the nucleotides are held together by weak hydrogen bonds. The molecule begins to unzip at those weak hydrogen bonds, and as it unzips, new nucleotides begin to fill in. Okay, so you'll start to see them filling in. All right, now when that happens, eventually what's going to happen is you're going to completely unzip the molecule. So you'll end up with the two halves completely separated from each other. And notice I'm using color, and I'm using color for a reason so that you can see what happens here. When the process is complete, you'll see that all of the nucleotides are identical all the way down the line. Okay, so notice here the A and the T, A and the T, T and the A, C and G, and so on. Okay, so you end up with two identical pieces of DNA. So we've effectively copied it completely. Now, the other reason for the color is because I wanted you to recognize that DNA replication is semi-conservative. Okay, semi-conservative. You'll notice that the black pieces of DNA, the half of the new molecule, that came from our original strand back here, right, as it unzipped in step 1 and then in step 2 when it unzipped, right? That was the original piece of DNA. And you'll notice that each new DNA that we create here, each copy, has one original piece in it, so we're reusing half of the DNA, plus the red, which is the new piece of DNA. Okay, so this is semi-conservative because each of the new copies of DNA contains half original and half new DNA. So that's why we say that. Also, remember what I said before. Even if it is not explicitly stated, everything inside of cells is controlled by enzymes. So everything here is being done by enzymes. The unzipping process that takes place at 2 and 3 is enzymes. Enzymes are filling in these new nucleotides that you see on the new molecules. Okay, so all of this stuff is controlled by enzymes, and this process takes place where the DNA lives, and the DNA lives in the nucleus. Okay, so DNA lives in the nucleus. That means this process of replication has to happen in the nucleus, and it is controlled by enzymes. All right, the next thing that I want to look at, that's replication. That's really how it happens. The DNA just kind of unzips, fills in, you're good to go. Now what I'd like to look at is to put this DNA stuff into a little bit of perspective for you, and to do that I want to do that with the genetics hierarchy that you see here. And this is also in your notes. So we are looking at the hierarchy of genetics. Remember, hierarchies mean as you move down the list, something is going to change, and in this case, this is going to get smaller as we move down our list, okay? So the hierarchy begins, the largest part of your genetics is your genome, which is all of the chromosomes. I'm going to run out of room here. Yes, all of the chromosomes are your genome, okay? So the genome is made up of chromosomes. Chromosomes, you'll remember from the last unit, are made up of coiled up chromatin. Chromatin is the DNA, which is written in a series of genes, and genes are written in something called codons, which we're going to get into a little later on. And codons are made up of 3 nucleotide sequences. Okay, so this is kind of helping you to put in perspective some of the terms that you're about to see coming up and how they relate to each other. So the biggest is the genome. Genome is made of chromosomes. Chromosomes made of chromatin, and so on. Another way of thinking of it is this way, okay? And this is kind of reversing the hierarchy from the next page. So we're getting smaller here as we move up, okay? Nucleotides make codons. Codons make up genes. Genes make proteins, and proteins make traits. So that's just another way of thinking of it if the first way confused you.